Dane, can you hear me? Can you say something if you
Yeah, I
can
can
hear
hear
me?
you fine.
Okay, I'm going to use this Bluetooth microphone, so you should be able to hear me even if I walk away from the computer.
Okay, sounds good.
Yeah, okay.
And I don't train with the track team either, so I'm like, I dodged like 3 bullets and half of my force.
Well, we both did. It's not even fair. Well, I really hope she crushed the trick. No, but I had, well, there are people keeping these in the room, and no one on the track team tested that.
Yeah.
What's the buzz?
Yeah, so there was like 3 or 4 kids in my floor that are gone.
Yeah, I really.
Austin, right. You guys need 6 feet because every time I get freshman year, I just put it on.
Very good. No, I got it.
Oh.
My grandma was very. We did it for a really long time.
Okay, gang. In order to conduct tomorrow's lab, you need a reagent called EGTA. I ordered it 10 days ago. It's still not in. No buffer, no reagent, no buffer. No buffer, no beads. No beads, no column. So I think we're going to have to cancel lab tomorrow. They emailed me and said, it's been shipped. You'll have it by tomorrow. Well, when tomorrow? Well, no, it won't be 10:30 in the morning. And then by the time M&D gets it, alerts me, gets it over here, it'll be 2 o'clock. That's assuming it actually gets here on time, which is itself a suspect assumption. So we're a little ahead on lab as it is. So we don't have lab on the 27th either. That's okay. We'll just finish up lab in November. We'll still be done well before Thanksgiving. We'll be fine. All right, so no lab tomorrow, sleep in. Your lab reports then, since you'd be turning them in tomorrow, we could make those due on Friday then. Now, we're having some trouble locating some of the old papers that were put in your lab. Those used to be readily available on PubMed, and they're not anymore. So I have a copy of one paper. I put it on Blackboard. Some of the JBC articles are available. The others aren't. And so I've done a search, and hopefully I'll get copies of some of those. If I do get them, I'll put them online. Okay? All right. There was also a request that the test next week be moved to Wednesday of next week rather than Monday. I've agreed to move the 404 test to Monday, so I'll go ahead and move this test to Wednesday. That would be the 28th, I think, yes? Okay. Let's see, we finished. We finished, I think, chapter 3 last time for our last, is that correct?
On the series, it says from meeting 12 to 21. So that-
Well, that's assuming we're on schedule, which I'm sure we're not.
I know that.
Yeah, so this test then will include chapters 4, 5, 6, and 7. 4, 5, 6, and 7. All right? Andrew?
I have another question, not about the exam, but what are the upcoming projects that COVID paper that was?
Yep.
How do you suggest going through that?
Slowly,
But like,
deliberately, and in sections. Take it section by section, because the paper builds upon itself. If you're having trouble visualizing what is there, try drawing, making some models, drawing some pictures. You can make models. That often helps with the 3D part of it. You have to try and think 3D. Not always easy, but biochemistry these days is very structural and requires a lot of 3D thinking. So you do it bit by bit. It's kind of like how you eat an elephant, one bite at a time. Okay. Yeah.
So since it's just the one article, it's still 6 pages.
Yeah. They used to have summaries of articles. They didn't have an article summary for that one. So, oh well. Say love you. If you think a figure helps you get your point across, then use figures. If figures would simply distract from the point you're trying to make, then leave them out. I had a student one year who fancied herself an artist, and so handed in a summary of 4 drawings, saying, here's what the paper's about. And they failed that assignment, because that's not what it asked for. Anyway, so that's the up and the long and the short of that one. Okay. We have lots of quarantines. So the guys soccer team's quarantined, the gals soccer team's quarantined, and that's the way it goes. My nursing class this morning was supposed to have 25 people in it. I think there were 8 people who were there. Everyone else was quarantined. Yeah, way it goes. So there we are. Okay, subject for today are biological membranes. Membranes are a phospholipid bilayer for the most part. This is nicely illustrated with this electron microscope picture. You can see very beautifully here, there are 2 layers to the membrane. There's a so-called outer leaf and an inner leaf of the membrane. Membrane composition was very highly debated for years. When you look at high school biology textbooks, from last century, you see all kinds of very interesting summaries of theories of what membranes look like. Turns out the people who figured out the structure of membranes were physical chemists, because people got very good at isolating membranes, telling you what was in them, but as far as how they were organized, the physical chemists were the ones who were able to say, this is the structure that thermodynamically makes the most sense. And then they took that theory and they applied that theory to relatively simple experiments and showed this is why the membrane had to have this structure. So it was two physical chemists, Singer and Nicholson. So it's called the Singer-Nicholson model, or the fluid mosaic model, but it's Singer-Nicholson fluid mosaic model. And if my memory serves me right, I think Singer and Nicholson won the Nobel Prize for that work. So, no, not the last decade, but the story of how membranes are put together, there's lots and lots of data. There were papers on compositions of membranes, paper after paper after paper. JBC, go back to the 60s, 70s, you'll find all kinds of papers about honeybee, membranes from honeybee gut have this composition of lipids, these fatty acids, these proteins, so on and so forth. Well, how's the membrane structured? Well, we don't know. And that was kind of the way it went. And then once Singer and Nicholson put together their theory, it all came together. That's how all membranes were organized. And it was Singer and Nicholson that did that. So the composition of membranes in terms of proteins, in terms of lipids, and what have you, they vary, and they vary wildly. In fact, even the inner and the outer leaf of a membrane can vary substantially. So here's some gee whiz numbers for you. So the lowly bacterium E. coli, lots of protein in this membrane. Lots of phospholipids, not very many other lipids, no sterols. Mouse liver cell membrane. Protein, but not nearly as much as E. coli. 30% phospholipids, 20% sterols, mainly cholesterol. Now, if you were to look at me and tell me with a straight face some 50 years ago that these two membranes had the same structure, you'd have been laughed at. But it turns out they do. Despite their respectable differences in composition. So here's compositions from membranes from plants, yeast, Paramecia, human myelin sheath, E. coli. Protein content differs pretty wildly. Phospholipid content differs pretty wildly. Sterol content differs wildly, and the types of sterol differ pretty wildly. Then you've got these weirdo other lipids, plasmalogens, what the heck are those? Galactolipids and all that stuff. And yet, they have the same structure. Here is a nice little pictorial way to illustrate this. So if this is cholesterol, and this is cardiolipin, these are minor lipids, sphingolipids, and two phospholipids, phosphotylcholine, phosphorylethanolamine. right then and there, you can see stark differences. So there's lots of cardiolipin, as you can see, in the outer mitochondrial membrane. Not quite so much here. More of your phosphatidylcholine here. Here. Phosphatidylethylamine varies, and cholesterol, pretty substantial difference. This is just within the same cell. So the composition of membranes just inside one cell can vary pretty wildly. So they vary between organisms, they vary just depending on where they are. So membranes have to have some fluidity and some stiffness. And they have to have that fluidity and stiffness across different environmental conditions.
So what do you mean then by they're especially similar if their composition varies?
The way they organize those lipids and proteins are virtually the same. So think of this. Let's say you're E. coli and you're living in, say, soil. Well, you have to be able to function if the sun hits you. That soil heats up to 80, 90 to 100 degrees. But how about last night when it got down to the low 30s? Your membrane still needs to be able to function. How do you do that to the membrane so that the membrane can have that nice mix of stiffness and fluidity? Animal cells solve that problem with sterols. Sterols stiffen the membrane. Bacteria don't make sterols. Plants use sterols, but the amount of sterol is very low. Also, they don't use cholesterol. They use stigmasterol. So how do we do this? How do we adjust for the firmness problem? Bacteria do it by varying the ratio of saturated to unsaturated fatty acids. Because remember, saturated fatty acids are nice and straight. They stack very nicely, very rigid. Unsaturated fatty acids have a bend in them. That's going to cause them to hack much less regularly. That's going to make the membrane much more fluid, not as stiff. So if you look at the fatty acid composition of the membrane lipids of E. coli over various temperatures, you'll notice, keeping in mind these are saturated, these are unsaturated, that your ratio of unsaturated to saturated fatty acids differs pretty wildly as you change temperature. Just look at this, just look at this. At higher temperatures, you have less unsaturated fatty acids. At low temperatures, you have quite a bit more of your unsaturated fatty acids. Here, your membrane has less thermal motion. It'll tend to stack more. If you have more unsaturated fatty acids, your membrane gets too stiff. Here, you have more thermal motion. If you have more unsaturated fatty acids, now your membrane won't be stiff enough, and the membrane will start to leak. So you down-regulate the number of unsaturated fatty acids relative to saturated fatty acids. Plants do this as well, even though they have sterols. So animal cells use sterols, yeah.
Will we be studying the mechanism at which they change the fatty acids?
No, I wasn't going to go into that, but I can mention just very quickly that the enzymes, Bob Nunn at UC Irvine actually figured this out. So the enzyme, the saturase, that puts the double bonds in to these, so that initially fatty acids are all made saturated, and then they are desaturated by desaturase. That desaturase in E. coli is not active at high temperature. As you go down in temperature, it now becomes much more active. Others do it at the level of gene expression. And there are some gene expression different in E. coli, but for other cells, they'll use gene expression more than just enzyme activity. Coli does it mainly through enzyme activity.
Why did it become unsaturated with a decrease?
Because these stack, they become very solid. Now the membrane is not fluid enough. It becomes too stiff. You want it stiff, but not too stiff. It has to have some fluidity to it. So in order to prevent it from becoming too stiff, put more unsaturated fatty acids that have that bend in it. So your stacking won't be as regular, and you'll still have some fluidity in the membrane. So the fluid mosaic model essentially predicts that your phospholipids kind of form an ocean, and your proteins float in that ocean like a cork bobbing up and on the ocean, or like icebergs floating on the sea, if you will. And the portions of the membrane proteins that are exposed both to the outside and to the inside are hydrophilic, but the bits that are embedded in the membrane are hydrophobic. And extensive research on membrane proteins has not borne this out, it's borne it out in spades. So if you look at proteins embedded in the membrane, you'll note that they have regions that in fact are embedded in the membrane. Those bits that are embedded in the membrane often are alpha helices. Those alpha helices are hydrophobic, and they form a so-called transmembrane domain. And you can see these in the sequence of the protein. Other proteins associate with the membrane because they are covalently modified. They have lipid groups covalently attached to them. The lipid group embeds in the membrane while the protein stays near the membrane. So biochemically, the protein may act like it's embedded in the membrane, even though in terms of its geometry, it's actually positioned close to the membrane, but not embedded in it. There. All right. So these others will have sterols attached to them and have you, but if you look at the sequence of this protein, you won't find a hydrophobic bit that sticks it in the membrane. Whereas with these, you will. Also, the chemical structure of phospholipids lends themselves to the fluid mosaic model. They have fatty acids that are hydrophobic, and they have a head group with a phosphate that's hydrophilic. So the hydrophobic part with the fatty acids sticks towards the center of the membrane. The hydrophilic part sticks to the outside. And these amphipathic membrane phospholipids, therefore, have two faces. The nonpolar ends face out and in. Sorry, The polar ends face out and in, the nonpolar ends face each other. And so if you were to take this and draw it with a sphere and then two stubby legs, the membrane would be arranged like this, with your fatty acids sticking in this direction, your phosphate and head group sticking like this, and this is the sea in which the icebergs of the proteins need to float. And from this, you can probably see why a set of phospholipids with unsaturated fatty acids tend to not be as ordered. They're going to have kinks in them and things like that. Sterols will tend to insert in between these, and also will be like in the picture that we showed from the book. These are like little sausages, if you will, stuck in one of the leaves of the membrane. Biological membranes have directionality. This directionality is due to the fact that the inner and outer leaf will have different compositions lipid-wise. You should expect that because there are processes that occur on the inside of the membrane that don't occur on the outside, and vice versa. So this, yeah.
Sorry, I'm not sure if you answered this already, but are sterols hydrophilic or hydrophobic?
They're both, because I think I have a slide with a picture of it. There's a hydroxyl group that's rather hydrophilic. The rest of it is sterile nucleus, which is very hydrophobic.
That's how they can be floated around
Yeah.
there.
Yeah. The composition of the inner and outer leaves differs, and in fact, it differs significantly. So some of these particular lipids, like sphingomyelin or glycolipids, they're found in the outer leaf. They're not found in the inner leaf. The inner leaf looks like phosphatidic acid, phosphatidylinositol, and it'll have more phosphatidylserine. That's important because very often phosphatidylserine is used to determine if a cell is too old. If that cell is too old, it is retired. And it is gauged by the amount of phosphatidylserine in the outer leaf. This shows you that asymmetry in the inner and outer leaf. So we can see here these two are enriched in their leaf. These three here are enriched in the inner leaf, as are these two, and what have you. So what I'd like to do now is show you some of the lines of experimental evidence that Singer and Nicholson used to confirm their theoretical considerations that provided them with the fluid mosaic model. And again, these are relatively simple experiments, but they confirmed their theory. So if you take lipids from a membrane and extract them, that's not hard to do. How might you do that? You could. That wouldn't be very clean. Chemically, could you use it? Sure. You could use ether. Ether would be one way to get your membrane lipids pretty nicely. So if you take those lipids and extract them to chemical purity, chemical purity is relative, it's a mixture of different lipids. Now you start to add those to water. here's what happens. At low concentration, they don't want to be around the water because water has to make clathrates around this fatty acid. That is entropically disfavored. They will therefore form these spheres that have the hydrophobic bit on the inside. The inside will be devoid of water. The hydrophilic bits on the outside. This is entropically favored. Now continue adding lipids to the water. And if you do, these will fuse and eventually they'll form a bilayer. That'll go across a container from one end to the other. Again, this makes thermodynamic sense. These fatty acids want to be with each other. They don't want to be with the water. So they associate together. And they put these ends together, these ends to the outside. Structurally, this looks a lot like a lipid, a membrane, doesn't it? So this will form all by itself. Then, as you increase the concentration of the water, of the lipids, excuse me, now you start to form an artificial sort of cell called the liposome. There's an aqueous cavity in the center, but that's okay because this is surrounded by charged elements that favor the water. These still are not exposed to the water. So essentially, this will form a membrane all on its own. Excuse me, if you keep putting it in the water at higher and higher and higher concentrations.
So could you separate lipids using ether because they're both hydrophobic?
Yeah,
That's the reason?
yeah. And that's pretty much how Singer and Nicholson did it. They just did ether extracts of various cells. So this told them that their calculations were in fact spot on because this was the structure that it would form all by itself. Second bit of evidence was that individual lipids show a surprising amount of movement in the membrane. The way they showed this was by attaching radioactive tags to lipids. That tag would be made in a glob of the cell. Then they would wait and expose those cells after periods of time to something called autoradiography. You start out with a glob, and then gradually those lipids will disperse throughout the cell, and you just have low-level radioactivity throughout the whole cell. That tells you, laterally speaking, the lipids have enormous motility. Enormous motility. Secondly, lipids can move from one leaf to the other, but you'd predict that they do this much less often, and they do. Lateral movement, very free. Vertical movement, not so free. This flip-flop, there's actually an enzyme called a flippase. Flipase. Flippases are found in virtually all organisms, and they flip lipids from one leaf to the other. This will occur by itself, but it's very, very slow. T1/2 is days. Oh, very good. Very, very, very good question. What's the point of it? What do you guys think might be the point of it?
lipids on the outside layer are downward on the inside.
Yeah, also, where do you make them?
On the inside.
So you're literally going to get uneven, aren't they? So to be able to flip them. That would be one of the reasons you'd want to do that. And this shows you the activity of the flipase. It now takes this flipping motion and now makes the half-life in terms of days. So lateral diffusion very fast, vertical diffusion is very slow. Enzymes, of course, speed that up.
When it flips it, is there a gas that's from it?
No, no, it fills in immediately.
How does it move across that?
How does what move?
How would a lipid, I understand how it moves from the inner layer to outward, how does it
Think of, you ever been to New York City? You ever been to Times Square? It's a mass of people. How did you move among that mass of people? Yep, that's how the lipids do it. They meander. Today, you can show this in real time using a technique called FRAP. Sounds like a dessert. It stands for fluorescence recovery after photobleaching. And you take lipids that are labeled with green fluorescent protein. So your membrane is green essentially. You zap it with a laser. And so you photobleach it in a hole in the membrane where there's no fluorescence. And then you could do timed video. You can watch the lipids fill in and the glow comes back. And you can time it and you can measure how fast the lipids move based on that sort of videography. It's very cool. Turns out proteins in the membrane are also quite mobile, which you'd also predict based on this model. So here's our old buddy FRAP. And FRAP, as you can see, you take those lipids or proteins, label them, map them with a laser, and then you can actually trace and time how long it takes essentially for these things to fill in. And they fill in very quickly. And then another piece of supporting data is that you can use agents that will label proteins. One of them is permeable. One of them, however, is impermeable. So these two reagents, as you can see, they will be able to form derivatives with the amino terminus of proteins. And if you do this, you see, you get two categories of proteins. The permeable reagent here labels pretty much everything. The impermeable reagent is only going to label those proteins that it has exposure to. That is, those proteins whose amino termini are on the outside of the cell. Those proteins that get labeled by the permeable one, but don't get labeled by the impermeable one, those are proteins inside the cell, intracellular proteins. They have nothing to do with the membrane. That impermeable reagent can't label intracellular proteins because they get through the membrane. But those proteins that are labeled by both, they occupy an external portion of the cell membrane. And again, you could label this as radioactivity. Now, proteins can be integral or peripheral. Integral or peripheral. Yes? They're listed as radioactive dyes, but they showed you membrane protein bits that were sticking outside. Bits were specific. So you weren't just throwing it up there. And the protein was inserted into the membrane in a very deliberate way. That also was predicted by the Singer-Olson model. All right. If the protein is integral, you have to dissolve the membrane to extract it from the membrane. So you can see here, this protein here is integral. Use very strong detergents, you'll be able to isolate it, it's integral. The only way you extract it is to do it with the membrane. Peripheral membrane proteins have associations with the membrane, but they are not of a nature that embedded in the membrane. So you can extract this from the membrane without destroying the membrane. You can use gentler detergents and isolate peripheral membrane proteins. So you can use things like Triton X100 or Tween or something like that. Cetramide. Gentle proteins is where this use things like SES or CHAPS or something like that. So those are your integral and peripheral membrane proteins. All right, so integral membrane proteins, you can remove them if you do away with the structural integrity of the membrane itself. They're held there by hydrophobic interactions. They will contain either one or more hydrophobic alpha helices. They come in these different types, as you can see here. Type 1 and 2 have only one, and one, as you can see, have the carboxyl terminus on the inside, amino terminus on the outside. Type 2 are switched. Amino terminus is on the inside, carboxyl terminus on the outside. Type 3s, you have a multi-pass protein. Type 4s, you have separate polypeptides that associate with each other to form this structured protein. A lot of pores are this way. That essentially forms some sort of complex. The membrane. Type 5 acts like an integral membrane protein, but it's there because there's a lipid modification. Type 4, both the hydrophobic alpha helix and like lipid groups as well. That's your type 6. This is one polypeptide. These are multiple polypeptides. G protein coupled receptors would be type 3s, but acetylcholine receptors, nicotinic acetylcholine receptors would be type 4s. Peripheral membrane proteins, as you can see here, this is the photosystem of Rhodopseudomonas. This has integral membrane proteins and peripheral membrane proteins too. That can be stripped from the complex without the membrane going kablooey. Nice British word there, kablooey. Lipoproteins have lipid groups attached. The lipid group is embedded in the membrane, so the protein has behaviors that are integral membrane protein-like. There's several different types of these. Many of these proteins are involved in cell signaling. So you can have a cysteine that's attached to usually a palmitoyl group. This is an internal cysteine, not a cysteine to the C-terminus. Myrisylation is at the amino terminus, and it's usually an amino-terminal glycine that has a myristyl group attached to it. The SARC oncogene has this modification. Farnesylation, as you can see here, is attached to a C-terminal cysteine. You usually have a sequence like this at the C-terminus. You have a cysteine with alanines and then a whatever at the C-terminus. When you attach the farnesyl or geranyl group, you cleave the protein right here. So this goes bye-bye. And then the carboxyl group of the cysteine is methylated. The reason you methylate it is so that you don't have a charge group right up against the membrane. This phosphate, that would repel this. So you make this neutral so it can be accommodated to the membrane very easily. The protein that is modified this way that has attracted the most attention is the external glycoprotein of trypanosoma. And these proteins are very heavily modified and they vary extensively, keeping our immune system guessing. No, I had that wrong. It's not trypanosomes. I'm sorry. It's another protein in the trypanosomes that's modified in this way. And the way they do this farnesylation is different than the way we do, so it might be a potential drug target. And then you have the GPI anchor, which I'll talk about in just a bit. This is a whole nother ball of wax. So here is the farnesylation that you can see. It's either a farnesyl group or a geranyl group or a dual geranyl group, depending upon what titillates your fancy, essentially. You see all of these, but again, they're all attached to a cysteine. That's the important part. GPI linkages. These are glucosyl acetylinosytol linkages. GPI linkages. So you have a acetylinosytol. So you have your glycerylphospholipid, your phosphate, and then the sugar inositol that's attached to it. To that, you then have a short oligosaccharide, and then you have your protein attached. Proteins that are linked to the membrane with a GPI linkage need to have a great deal of flexibility and movement. So for example, in the immune system, there's a protein on macrophages called MAC1. That's linked to a GPI by a GPI linkage. The T cell marker in mice, Thy1, is linked by a GPI linkage. And then so is the glycoprotein, external glycoprotein from trypanosomes. And that's an interesting little thing. So here is a GPI linkage. You have your phospholipid here. Here's your inositol. You then have this linkage to this short little oligosaccharide. You've got glucosamine, ethanolamine, phosphate, a glucan, a glycan of some sort, phosphate, ethanolamine, then your protein. This is generated in the ER. There's not a sequence patch that tells it to happen. Instead, there's a 3D structure of some sort that the GPI transferase recognizes. And it lops off the protein. This thing is pre-built. So it lops off the protein, attaches it to this, and C-terminus stays stuck in the membrane, essentially. And the rest of the protein is just there to do its job. And then it gets trafficked to the membrane that way. So that's a GPI linkage. Any questions about any or all of that? No? Very good. Let us continue.
Which direction was it? Is it on the inside?
No, those are stuck to the outside. Stuck to the outside, yeah. Those face outside. Right, okay, so share screen. This one. Very good. Signaling proteins very often localize to structures, substructures in a membrane called a lipid raft. A lipid raft. Lipid rafts for years were hot topics. They're still kind of a hot topic, not as hot as they used to be. They've cooled a bit, so it's like the interior of the sun. They're more like the surface of the sun. But every cell biologist wanted to study lipid rafts because wherever you looked, signaling proteins were sorting to a lipid raft, and GPI-linked proteins tend to sort to a lipid raft. So what is a lipid raft? A lipid raft is a portion of the membrane that is enriched for sphingolipids and cholesterol. So it is thicker than the rest of the membrane, because remember, the sphingolipids, they're longer than your glycerophospholipids. And those longer lipids with the cholesterol gives you this membrane now that has a increased width to it. And you can see various types of signaling proteins will sort to the lipid raft. And if you look at the thickness of a membrane using this atomic force microscopy, you can see these are lipid rafts. See how thicker these are than the rest of the membrane? That's an example of a lipid raft. That is a spike. That's an artifact of the machine. As you do this atomic force microscopy, you're measuring topography using this instrument that's very sensitive, and it gives you spikes occasionally. And that's what it does. Still a nice picture.
When cells are keratinized, they still have this like fluid bilayer or membrane.
That tends to dissolve because the keratin tends to take up everything. You also have other proteins that are made filigrees and other proteins that seal it. And it makes it very water resistant. If you take a shower, you notice the water beads off your skin. That's the oils, of course. Sebaceous glands, but the keratinocytes themselves are very water repellent as well. Divya? Slides are moving. Well, let's try this again. Okay, now they're moving. Jenna, if you unmute yourself and say something, I can actually hear you because.
Yeah, they're moving now.
Okay.
Yeah, they're
Ooh.
good.
Oh, that was loud. Turn that down. There. Ooh. Blow out those eardrums. Okay. Now, there are little pits you find on cells, and they're called caveolae. They're formed by these little proteins called caveolin. So caveolin, as you see, has these lipid groups, and this will form dimers. And when they do that, they deform the membrane to form a little pit. Why is this interesting? Interesting because these structures are required for regeneration of the liver. So in a rodent model of liver regeneration, you can actually take out close to a third or even 2/3 of the liver, and the liver will actually regrow. And if you get rid of caveolin, so they can't form caveolae, they can't regrow their liver. Also, when you look at caveolae, you will notice that they're filled with certain types of signaling molecules. Some signaling molecules specifically tend to map to these caveolae. So these areas, essentially, of membrane deformation may be areas where you have most of your cell signaling that occurs. A little penny for your thoughts. Membrane fusion is something that was first studied using viruses that are very similar to influenza. Now, influenza has on its surface an H and an N protein. H stands for hemagglutinin. And N stands for neuraminidase. These proteins had functions that were really not well understood. Hemagglutinin could take red blood cells and clump them together. So it was called hemagglutinin. Neuraminidase would cleave sialic acid residues off glycoproteins. So it was called neuraminidase because it cleaves off neuraminic acid, another term for sialic acid. Turns out these proteins, when you get inside an endosome, the pH of the endosome now starts to decrease. These proteins form trimers. When you decrease the pH, the amino terminus of these proteins, which are normally embedded, the protein does this twist, and the amino terminus will do this. When you drop the pH, now you start to protonate residues that repels this, and this thing goes like this, and this piece is hydrophobic, so it sticks to the membrane. Then these particular alpha helices here will form a three-fold coil structure. What does that do? It takes these two membranes and it brings them so close together that they have a choice. Either sit there with repulsion and deal with it, or fuse. Fusing is thermodynamically the more conservative approach. So the membranes will fuse, and by fusing, now the virus will escape into the cytoplasm. And by escaping into the cytoplasm, it can now liberate its nucleic acid, start to replicate that nucleic acid, start to translate the viral RNAs, make viral proteins, and infect the cell. And this is very similar to the way other proteins in our cells work. And you can see that this fusion is very common in our cells. These are called snares. Snares. And this is a snare found in a virus. We have very similar proteins. They're called snares.
How does the endosome
It binds to the receptors, and that stimulates receptor-mediated endocytosis. This then fuses with a lysosome. pH drops, then you get the conformation change, and then that gives you the HA fold, what have you. And it looks like our time is gone. Okay, we will pick this up on Wednesday. Sleep in tomorrow. You're welcome. And
Do we have a day that we're making up the lab?
there's no, yeah, we'll make up the lab the first Tuesday in November.
Okay, so the 20 oh, so the 3rd.
Okay,
Okay, thank you.
you're very welcome.